characters. Your book is fine, and this is terrible because I work on plants, but this is trying to show the same thing, but I think the bunnies are a more effective way of getting across features that can be used to build a phylogeny. And that's what this is trying to show. This is from your book, but it's very similar to the bunny example. But and again, it's using characters. The color of the petals would be a character. The colors would be the states. It's using features of the stem, whether it's herbaceous or woody, as another character, and you're looking at states of those. So you can look at that, but then to get the idea. But then I have the link in here. I think it was there for that Tree of Life little video, and it's trying to do the same thing. You can watch it yourself and go to the end and see the whole thing. But does everybody get the idea of what we're trying to do here? We are taking characters, characteristics, features, flower color, stem structure, and then you have to have states for each of those. So for example, if it's flower color, it could have several states. It might be white, they might be red, they might be blue. Those are actual states. Or stem structure, woody versus herbaceous. The bunnies, what was the characteristic there? Color, yeah, and then the states were purple, red, and tan. And that's what you're going to do when you build phylogenies. You need characters and you need states. Good? All right, and then you can also see how these things evolve across the tree. So we need characters and character states, and we're going to reinforce that in lab. So if this is a hypothesis, just like the bouncing bunnies, if you start with an ancestor and then there's a mutation in the flower color there, you're getting a new species evolving, and maybe you're getting a new species evolving here, you're able to pick those up by looking at these characters and these character states. Again, this is from your textbook. It's a little complex, but at the end, what you're ending up with in that example is something like this, where you started with an ancestor, okay, and then here we had the ancestor had white petals, herbaceous stems, and then it added other characters. The leaves were non-hairy, the stamens were fine, the fruit dry, the seed coat smooth. Well, here there's a change in color to petals red. Oh, and then there was another change over there to change the stamen number. Oh, and another change here. All of these are involving different characters and states for those characters. Everybody okay with that? All right, this is just a little bit more involved. And then there's a change from this herbaceous to a woody stem. Oh, and now the change of the fruit from being dry to fleshy. And the seed coat is now spiny instead of smooth. That is a phylogeny. And that is being constructed using characters and character states. That's how you build trees. That's how you build phylogenies. Now we can also use some of our terms, some new terms, to look at that phylogeny. We're just now taking the very same thing and introducing some new terms that you're going to see and use in the class. One of the most important terms, probably the most important term up here, is something called a synapomorphy. It's a lot, that's a big word. But use it, embrace it, you'll hear it a lot. A synapomorphy is a shared derived character state. See how we can talk now? Like 10 minutes ago, you didn't know what a character was or a character state. Now we're showing you how you can use those to build trees. A shared derived character state is a synapomorphy. So here's a great example. Here was our ancestor. We talked about this change in flower color from white to red. Both of those species have red flowers. You see that? Right here. This is a change that occurred on that branch of the tree. You'll see this a lot. Where a change occurs, you use a little slash, and then you put the change. The petals are red. The petals started out white. There's a change to red. What is that for those two species? It is a synapomorphy. It is a shared derived feature. It's not in their ancestor. It's shared by both of them. It's derived. That's why we call it a synapomorphy. Same thing over here. In this group of plants, the stems started out herbaceous, but they changed. There was a change, an evolutionary change to a woody stem. Both species 1 and species 2, we're not putting names up here, have that feature. So what does that make? That character change right there is a synapomorphy. Everybody see how we can use that? You're going to hear this over and over again. So let's go back to the, oh yes?
It only applies if it's within a monophyletic group.
Exactly. Synapomorphies are features that will unite a monophyletic group. Synapomorphies define clades. You set that one up perfectly, because if we go back to our phylogeny that I showed you for green plants, you remember that? I said the uniting features for green plants were what? They have chlorophyll A and B. What was that? Starch, whiplash, flagellum. So those must be, using our newfound terminology, fill in the blank, those are synapomorphies for the green plant clade. Whoa. Okay, so what have we learned? We've learned about clades, which are the same thing as monophyletic groups. We've learned how we're going to define those. We're going to define those using characters and character states. And a character that unites a clade is a synapomorphy, a shared derived character state. That is, they all share it. It's derived from an earlier ancestor. That's a synapomorphy. Does everybody have that? Very important, because throughout the class you're going to hear, okay, we're just going to be throwing that around all the time. What are the synapomorphies for the land plants? What are the synapomorphies for the flowering plants? And what those are will be the features that actually define those as clades, that make them recognizable or diagnosable as clades, as monophyletic groups. Everybody okay with that? So I know it's a lot of terms, but this is like foundational. We're going to use, now if you know these, you're going to use them a lot. We'll use them a lot. Now, notice that species 1 is the only species that has that fleshy fruit, fleshy, it's hard to say. So that's what's called an autapomorphy. Aut just meaning one. It's unique to that species. So it's a uniquely derived character state. You'll see that term in your textbook too. Autapomorphy, synapomorphy. This is the biggie. This is the biggest, most important one. Sometimes you'll see this, the ancestral state. These are the ancestral features, and those are called simplesiomorphy. So the petals being white, that would be the ancestral state. That's a simplesiomorphy. But how we define clades, what we use to build our trees, the most important things are synapomorphies. Those are going to be the things that unite groups of organisms. Good? All right, that definitely is something, think about that tonight. We're going to use that term a lot. All right, so let's look at it. This is from your book. This is a simple phylogeny. Each one of these would represent a change, a character that we were using to build the tree, okay? So there are 3 species, Roman numeral 1, 2, and 3. 9 is what? Using a sentence, as I just introduced terms, what is 9 for what? Well, first of all, what are 1, 2, and 3? Together they form a clade, or a monophyletic group. Do 1 and 3 alone form a clade? No. Do 2 and 3 form a clade? Yes. So 9 would be a synapomorphy for the clade 1, 2, and 3. What are the synapomorphies for 2 and 3? 3 and 5. Okay, those would both be synapomorphies. Okay, I don't know why I have an arrow at 5, but maybe that'll come back to me. I don't know. Okay, and I'm just showing you. This is one of the synapomorphies. And then, of course, 3 would be the other synapomorphy. Now this, 1, 2, 4, 6, 7, and 8, those features that are only present in 3, those are autapomorphies, right? They're just unique to that one species. So that would mean 2 is what? An autapomorphy for 1? Got it? Yes?
Would 9 be an ancestral state for the clade 2 and 3? Like if you were just looking at 2 and 3 as a clade, would 9 be an ancestral state?
It would be, I guess you could say, is that symplesiomorphy for 2 and 3? You could use it that way. Yeah, that would be the ancestral features. That's okay. Yeah. We probably, that's a good point. You know, sometimes these terms are kind of relative. So say that a new species is discovered, and we'll call it species 4. And it turns out, we can even use our terms, it turns out to be the sister of 1. So it branches off right here. So then 2 is what? Now it's a synapomorphy, because both of those species have 2. So if we discover a new species, it goes there, now suddenly 2 could become a synapomorphy. So keep in mind that these terms, and you just mentioned, that was your question, it shows the sort of the relative nature of these sometimes, based on new knowledge and new species and how they fit into the topology. Okay? Everybody go back and review some of these. I know it's a lot of terms, but they're very fundamental. Okay, let's go back to this. Here is our phylogeny for green plants. All green plants together, all 500,000 of them form a, what are they? They're a monophyletic group. They're a clade. We're only going to focus on clades. We like to talk about clades. That's what we like to name, clades. Not groups like reptiles that aren't clades, that really a clade. We focus on clades. So if you saw this phylogeny, this branching pattern of how things are related, you could say that is just the branching pattern itself, if you wanted to talk. The topology. Okay, so, it's an interesting feature in this topology that, like here mosses are next to tracheophytes or vascular plants. We'll get into that later. If you're just talking about aspects of the branching, that's topology. So these features, anterior flagella, starch, chlorophyll A and B, are what? Use them in a complete sentence. For green plants, they are the synapomorphies for the green plant clade. Everybody have that? All right. So there are, thousands of species of tracheophytes. So the presence of tracheids and branched sporophyte, even if you have no idea what those are at this point, must be synapomorphies for tracheophytes, okay? And here are all the land plants. If I had to ask you, if you were given this, and I asked you, what are the synapomorphies for land plants, you would say cuticle, archegonia, and antheridia, these are reproductive structures, an embryo, and a thick-walled spore. Are you getting the idea of how we use trees, how we use phylogenies, how we use the term clade, how we use characters to build trees, and the idea of a synapomorphy? All right, a shared derived feature. Got good? All right, I think that sets us up pretty much for a lab. I will also say that DNA is a hugely important character. Characters and character states. We're building trees. We'll get into that next time. And other aspects of building trees, but we're out of time. Okay, good. All right, well, so we'll see you in lab later on this week. If you have any questions, please don't hesitate to let us know. All right? Good, anything else before we go? All right.